everyone. I'm Emily Cranston, and it's my great pleasure to introduce to you today our BPI seminar speaker, who is Professor Eleftheria Roumeli from the University of Washington. And so Eleftheria is an assistant professor in material science and engineering. She did her postdoc at Caltech and ETH Zurich in mechanical engineering, and her PhD and bachelor's degree are in physics from the Aristotle University in Thessaloniki in Greece. And as you may have guessed from her name, it is also Greek. And I can say that Eleftheria and I met a few years ago, but full disclosure, we keep giving talks at conferences at exactly the same time. So we haven't heard each other speak, but we've seen each other's students, and we're very aware of each other's work. So I'm really excited to finally get to hear her present her work today. But also, since meeting her, I was on vacation in Greece, and I saw her name all over the place, and I thought, oh, this word is everywhere. So I looked it up, and it means freedom, which is lovely. And so the word appears lots of places. You'll see it if you go to Greece. And I thought that freedom is actually a really fitting term, because on the good days, academic research is a great representation of freedom, so on the good days. So Eleftheria's current group explores new families of bioplastics, biocomposites, and environmentally friendly construction materials that are derived from biological building blocks and specifically from biopolymers. That's obviously what she's talking to us about today. She has been the recipient of a number of awards, especially for early career researchers. I won't list them all, but in 2022 and 23, she was the University of Washington Materials Science and Engineering Faculty Member of the Year. She received the 2024 ACS PIMSI Early Investigators Award, and she was selected as a Kavli Fellow for the 5th Korean American Kavli Frontiers in Science Symposium in 2024, and she has a 2023 U.S. National Academy of Science Young Observers Award. And so congratulations for your awards and everything you've done so far, and thank you for taking the time to come and visit us here at UBC. Please join me in welcoming Eleftheria.
Thank you. Thank you, Emily. This is way too kind. Half of those are from students, so favorite teacher, you can say. That means something, though. Okay, so let's see. Hopefully you won't be too disappointed that, with what I brought, and you won't feel that you will feel that you have missed out on the previous ones. Let's see. I only have one slide for general motivation because actually you guys are motivated in your own research in your institute by pretty much the same things. So the main motivation is the issue around sustainability. Maybe 3/4 of our lab are working on designing new types of plastics, basically. So majority of the motivation comes from the things that we want to change around plastics. They are ubiquitous in our polymers in general. I will be referring to them as plastics, including thermosets and elastomers, just for the sake of simplicity. These are ubiquitous class, of course, of materials. We need them for our everyday lives, but they do come with a set of challenges that touch actually on each and every step of their life cycle. There are challenges in their sourcing, where even in Europe, when they're doing a slightly better job than the US, you see that we have a majority of our sources for sure are going to be from petroleum. Then we have challenges related to processing, where maybe you may think it's not the biggest deal, the emissions, for example, or the waste, or the types of solvents that you're using. Maybe you're thinking, as do I actually, that this is not the biggest problem compared to those two. But here is where we have the majority, of course, of the problem at the end of life. End of life is challenging for many, everywhere actually, and it has been inspiring different types of strategies to mitigate some of the damages that we already have done. Some of the things that people are working with, including people in your own institute, of course, are optimizing for a non-harmful, for a harmless end of life. Benign end of life. Other people are working on strategies for improving the collection and recycling processes. All of those are solutions to the same problem, I guess, from just different standpoints. So if we break down in 3 groups, just a little bit of an introduction of what's going on sustainable polymers. I recognize that you probably know all of this, but I will have to go through some introduction. There's 3 different pillars that typically we want to be hitting. We want to be discussing that our source is going to be renewable. We want to see if our processing is going to be as green as possible. And of course, we want to have a manageable end of life. Manageable just to include also the recycling, because how do we manage that is actually a big problem. So majority of the work, especially from a polymer standpoint, by far, I would say majority of the work actually focuses on addressing this last pillar over here. A lot of work, especially we have catalysis people in the room, so a lot of work on using these beautiful catalysts that we actually have been using to make polymers now to do the reverse, to depolymerize controllably. So we have different examples. I only chose to highlight one here just for giving an example where we have mixed olefin waste and we have a cobalt. This is work from a colleague of mine, actually at the University of Washington, Professor Julie Rohr in chemical engineering. It's work from her postdoctoral studies where they designed this catalyst where it can selectively depolymerize mixed polyolefin waste into light alkanes, and then they show different yields for their processes and different kinds of mixed feedstocks over here. A lot of work in that domain. Then there's other processes that are not relying on catalysts. This is an example where we have thermal it's a pyrolysis-based process, so you have basically a thermal gradient. You put in, again, mixed polyolefin waste. You have this column over here in which you have a controlled temperature distribution, and you go under saponification process where you are generating a very lightweight waxes, basically, that in this particular work, it's quite seminal, actually, because they have demonstrated that you can actually make wax that you can use afterwards as a soap from literally mixed plastic waste. So I think it's very nice heat-based and catalyst-based processes. Of course, there is a significant work on addressing also the sourcing element as well. So going now to renewable sources as well, so deriving monomers or oligomers from biomass. A lot of this work actually has been done here with lignin, for example, as well as other types of monomers. So we have seen that you can extract a wealth of very, very useful functional monomers or small molecules, and then you can use synthetic polymerization processes, and then you can balance. Now we are designing based on a balance between degradable and durable. So a lot of the impactful work doesn't stop from, of course, getting something renewable as your feedstock, but also how do you address this balance over here. A lot of this is actually a relatively older review. It's 2017, but I still think it's one of the most nicely, very comprehensive ones. And then there's, of course, again, designing for this end of life, either with renewable monomers or not. You have a lot of progress right now, of course, in dynamic covalent chemistry. So we now have a plethora of different polymers that are made with dynamic bonds that can be selectively disassociated with specific stimuli so that you can either recover your parts, or you can controllably break down some of them. So this is from a recent review that is covering what's going on that topic. In all of those cases, though, at best case scenario, you are deriving some monomer from biomass. So you are generating a lot of waste. You are creating waste streams. How do you go down to depolymerizing a whole high molecular weight polymer like cellulose, for example, or like lignin, doesn't come for free. So taking a step back, a lot of work. I should have changed this to feature who is doing what here, but I think you get the point. You have done like half of the literature in cellulose is coming from the people here. So it's not your work, but it could be. It's taking a step back to show that, okay, maybe we should be thinking about using this entire molecular weight that already we have. It is synthesized for us. Not just the molecular weight and the functionality sometimes, but also sometimes the crystallinity or structure or topology of those materials, of course, can be used. So you have examples like here where you just remove lignin and maybe some of the hemicelluloses so that you can have moldable wood in which basically, this is from Bing Ku's group, in Maryland, where, of course, you can take advantage of the natural alignment as well of cellulose within your cell walls. So you don't just take advantage of the polymer, but also of its conformation in space. Other examples is extracting CNFs or CNCs, and, of course, hydrodynamic alignment, and using processing now to create assemblies that will add to the properties that you inherit from cellulose. So all of those are addressing more and more of the design goals that we want to be hitting. But even in the case of cellulose, for example, or chitin, you still create some waste streams. You still need to extract this somehow. Now we have the eutectic solvents. You have other greener stuff. So definitely this gets better. The process gets better. But you are not utilizing 100% of the biomass with that strategy. In my group and others, of course, in literature, we are looking at taking a step even further back and trying to see how we can utilize the entire biomass. So in my this is a snapshot from my postdoc where we actually were culturing plant cells. This is undifferentiated Nicotiana tabacum cells. So it's this and Arabidopsis is the two model plant organisms if you're studying plant biology. So you can find these everywhere. So this is two cells, mother-daughter cell, that we were culturing in the lab. In the cell wall, they have a cellulose-reinforced plant cell wall, just a primary though. So it's actually, that one is not ordered. It doesn't have the secondary or the multi-layered secondary cell wall that maybe you would be used if you're studying wood. You would be more familiar with that. It only has the primary. Still, it does come with cellulose, phenolics, pectin, and hemicellulose. So we were trying at that time to see how, instead of extracting the cellulose or other polymers, how can we find a processing method that would allow us to use the whole hierarchical structure of the cell wall as it is without creating any waste. So in that particular case, we designed a compression molding approach in which we simply were following the loss of turgor pressure on a plant cell to induce compaction so that the neighboring cell walls or cell walls between neighboring cells that were being compacted, they had fibrillar interlocking, so there was a polymer chain diffusion in between them. There was also, of course, hydrogen bonding in between them. That was enough to hold the whole thing together. And then in terms of mechanical properties, that is a relatively stiff and strong polymer composite, if you will, that can be molded. You can do something similar to what you would do with a conventional commodity polymer as well. So this was where we started from. And all of the things that I spoke about in terms of literature, I guess, what has been done in literature, we did put last year a couple of reviews out. If you're interested, let me know. I can share them with you. So the whole idea of utilizing entire organisms, many people, of course, are working on that. We had started originally from plant cells. Right now, we are primarily focusing on algae and bacteria, but there's other people, of course, using significant, of course, work in mycelium-based materials. I think this is probably the most not well studied, but many people are working on it, and many startups in that area, bacteria, yeast, microorganisms, macroorganisms, and trying to understand what is it that we have, how can we make useful materials out of those. So then in our group, what we're trying to do in terms of, if we look at the mechanical property space, here's going to be an Ashby plot of a density and Young's modulus. You can see that in the blue balloon over here is the polymers that we derive synthetically, so from petroleum. In our case, we are aiming to use renewable, preferably photosynthetic biomass, and create solutions right here in the middle of this space, because here is our single-use plastic problem. So if we go out in our oceans or forests or even landfills, the majority of the plastic waste that we will find is going to be in that because it's the ones that you use the shortest of, and they have the smallest lifespan, and of course they're not degradable. So you guys are an innovating space up here, of course, very, very high stiffness, very high strength. For specific applications, you would need that. In our case, we're actually targeting the middle of the range for the reasons that I explained. So the work that we're doing, this is a map of the state of Washington, and I do challenge myself every time I show this to point to where Seattle is. It's somewhere here, but here is where we get our seaweeds, which you will see the results of. So this I do know, it's actually close to you guys. You must be somewhere up here, right? But look, it's on purpose. It's a very high level of difficulty. Okay, so the biomass that we are working with is typically something that we can find that is scalable. Either it is already cultured in the ocean or it grows by itself in the ocean, hopefully inexpensive. Some of it is actually waste, so we get it for free. ideally photosynthetic micro or macroorganisms. Because we want to be working with micro and macroorganisms, some of those are single-celled organisms, some form tissues and all of that, so you have also hierarchical complexity that you can play with, a building block versatility, both in terms of biochemistry and in terms of how your polymers are distributed in space. And then ideally, we want to be using resources that are locally available. So results that I'll show you come from one type of biomass, spirulina. It's a cyanobacterium. I think I have a couple of results from other types of macroalgae, actually. So I'm going to be walking you through that. So our first work that was on studying this single-celled organism that is a cyanobacterium, spirulina. It's a photosynthetic cyanobacterium. You will hear it also cross-listed as a blue-green algae. I'm not a biologist, so to me it doesn't matter really how do you call it, but it is a single-celled organism, so it's one of those, it's a single cell and does not form a tissue. So what we started off in the beginning was we have this that is harvested, as it's harvested from the pond. So we don't do any size classification. We do size analysis to see what are the size distributions. This is the SEM image of the dried powder cells. You will see you have very, very wide ranges in your sizes because these cells actually agglomerate and form chains that you can already see that they are very heterogeneous. And actually in biology still, to my understanding, they have not solved what determines how many cells are going to go in one chain. But nevertheless, in our case, we take it anyways dead. So what we do is we study the size distribution. We do, this is TEM so that we can see how the cell wall looks like, especially because my previous work for plant cells showed how important it is to understand the cell wall. Here, we don't even have cellulose. And you see that we're talking about a 20 nanometer, very, very thin and flexible cell wall that is rich in peptidoglycans. Then we go in and we start characterizing what we have. This type of unicellular biomass has about 55 to 65% proteins. This is just the pigment protein. Of course, we have different ones as well, but it is 55 to 65% protein. You have about 15% phenolics, low molecular weight, no lignins, nothing like that. It's just a small cell. 15% carbohydrates, both high molecular weight and low molecular weight. And others that don't fall in this category, you have also some inorganics as well. So you have the monomers over here from HPLC so that we can see that it's not, we're not talking about cellulose in here, even though we do have glucans as one of the monomers, but we do have a variety of other monomers as well. But definitely not cellulose. Here we see the molecular weight distribution from an SDS page so that we can see that the proteins have, each of these beans correspond to a different molecular weight classification for our protein. So you see we have anything from 15 to 70 kilodalton. That's because, so we don't extract anything, we just characterize what we have. But then you can imagine, because some of you I think work also with proteins, and in a nice, I guess, ideal case scenario, you would maybe have one of those, and it's going to be nice, and maybe dispersity of very, very little. Here is, I only put this here, not because it's a good measurement, it's not. If you actually do SDS page, you know that it's not a good measurement, but it is what we actually get. So I want people to actually see how the real data looks like if you put a real organism full without any pretreatment. So then, as a polymer physicist, what can I do with, I do not want to do any chemical reaction. So what I want to be doing is see how I can use processing methods to actually influence the way that these polymers are bonded or conformed maybe in space. So in the strategy that I lay down here, this is exactly what we would be using even for our polyolefins. You have some feedstock. In other cases, it could be your polyethylene. Here, it's just our spirulina biomass. You may need to put additives if you want to have a performance modification. The results that I'll show you have no additives unless I will tell you when they do. Then we put them in processing methods, like in this case, it's a twin screw extruder that you can see that it's extruding. It's a mixture of water and spirulina in this case. And then we also, if we want to make beams for mechanical testing or whatnot, we would have to also hot press different beams. So what we want to be seeing in this case is the SEM image of the hot pressed material and the SEM image of the material that we started from. And that's just from the application of heat and pressure. You can see that here you have a homogeneous mixture, basically, of polymers, but there is no phase separation. There is no... I would think that maybe the fats would partition out, but we don't actually... I mean, fats, you only have actually maybe 5 to 9% fats here. So nevertheless, you don't see anything like this. You see one homogeneous thing throughout. So we're trying to see now what is it and how can we reproducibly do that. So then our second PhD student, Harish, he started from a design of experiments where you could do iterative, but you can do design of experiments or even machine learning these days if you want to explore a variety of processing conditions. Here we were doing actually, in the example that I cited, it was an iterative, very small DOE, where we vary the pressure, time, and temperature in the hot press alone. So we're not doing extrusion here, just isostatic pressure, so that we can see in the processing window that you see over here. So it's a rather large window, 60 to 160 degrees Celsius, 4 to 75 megapascal, 15 seconds to 30 minutes. We want to see what are the changes that we can induce to this powder, basically. So we're going to be doing 3-point bending tests for this particular experiment. You see different curves over here, just force displacement, stress strain curves. And then you see the extracted strength versus time. So it's a 1D slice where we're keeping the temperature and pressure, and we're only seeing the effects of time. And we're seeing how the different pressing time is influencing the strength of the beams that we are able to produce. And you see a very an exponential type of behavior. We have a characteristic time, tau, that denotes to us when significant change has occurred. We don't know yet what this change looks like, but we do see that after a couple of minutes, you already have a steady state flexular strength in your results. We chose 5 minutes, which is actually longer than the characteristic tau time, because we wanted to have not just to reach the plateau, but we wanted also to have as small of an error bar as we could. And I hope you will be paying attention to the error bars here. They are not, I guess, as scary as I thought initially, because this is unfiltered biomass, nothing has been extracted. So we do have error bars, but I don't think they're pretty bad. Then, if we want to look at a 2D plot of changing temperature and pressure, so here we're going to keep time fixed at 5 minutes, and now we're sweeping a different temperature profile and in combination with pressure. And the numbers that you see over here correspond to the values of flexural strength. So I'm not showing you all the data. We have one representative stress strain curve for each of the temperature conditions so that you can see how rigid and the materials are. These are very, very brittle. But you see that we are mapping here our flexural strength, and even if we look at a specific row where we are keeping our pressure fixed, you see the effects of temperature where we have variability about 26 times. We can go from 1 MPa to 26 MPa just by changing the temperature between 60 and 160 degrees by keeping the time and the pressure the same. So then if we extract the data for modulus strength and toughness, and this is the work to fracture basically, not fracture toughness, you can see that we have significant ability to play with the properties that we get. About 8 times you can improve your modulus, about 21 times your strength. And I do want you to look also on the y-axis for a second, because you may think that these are very, very weak materials. These are weak for polymers, but they're not that soft and weak. They're not the squeezy category. We are talking about strengths all the way up to 25 megapascal and moduli 2.5 GPa. So that's quite, and 1% or less, actually, elongation to break. So we're talking about very brittle, relatively stiff, and relatively strong. I will say plastics now and bioplastics, because those do flow with heat and pressure. So now I allow myself to say bioplastics because they do have the plastic behavior. and thermoplastic behavior we will see in the next slides. So if we look at the morphology of those materials, this is going to be the poorly bonded sample, so conditions that gave us very, very poor mechanical properties and conditions that gave us the best mechanical properties. And we can already see, of course, big differences. In this case, you just have loosely packed powder. You can distinguish all the cells, and you can see this comes from a large chain, this comes from a very tiny chain, or even maybe single cell, and you can see that even though it was strong enough to be measured, so we're not talking about powder, you still can actually grab this with your hands, but this is very poorly bonded compared to this, where it's basically an amorphous blend of everything connected. This is just for the morphological, of course, point of view. Then we will go in with spectroscopic methods, so with FTIR and XPS, to start understanding what is happening to the bonding in the polymers that we have. So here we have the FTIR. The green is going to be our powder as it comes out of the pond, no treatment. And the blue and the yellow are going to be the poorly bonded sample that we saw before. So the blue corresponds to this condition, and the green the yellow corresponds to the well-bonded sample. So we can already see a couple of things. First, if we look at the protein area, and maybe the protein 1, the amide 1 peak over here, you can already see that your peak contour has changed, and you actually have almost two separate peaks, or we have actually a peak in the lower wave numbers indicative of more beta sheet formation. Then we do the convolution for the alpha helixes, beta sheets, and whatnot, and we can see that you do have a shift towards forming more beta sheets in the better pressed conditions, which you don't have here, though you do still have some difference between the poorly bonded and the powder. So we can say that there is something is changing in our protein. We can keep that, and we can keep it at that level right now for the discussion right now. And then we have, of course, another red shift in ROH bond. So we know that there are also changes happening in our hydrogen bonding network. We're not measuring hydrogen bonding from FTIR, just seeing that there is a small shift between the powder and the well-pressed material. Then we go in with XPS, and we do the convolution for the carbon and the oxygen peaks. And here we have the ratios between carbon-carbon, carbon-hydrogen, carbon-oxygen, carbon-nitrogen, and carbonyl bonds. And we see that between the powder and the poorly or the well-bonded material, basically our ratios change. So then from all of this, what we can say is that our hydrogen bonding is not identical. So there is some change there, even though arguably minimal. There is something happening actually way more significant in our protein domain because of the shape of the peaks. And then there are also some changes even in our covalent bonds, where we have changing ratios between the different bonds over here. So this is I really thought that it would have been way easier, but it is very complicated, especially if you think of our slide 1 with all of the characterization, where you have 5 different monosaccharides building up your polysaccharides. You have 5 bins in your proteins, where you have different amino acid sequences in all of those. Then you have some lipids that actually play a significant role, and it's all over the place. You cannot actually, unfortunately, to my surprise, we could not actually come up to a reliable conclusion. So then we set up, we decided to study the whole thing in a clean way. So we devised a model system where we're trying now to simulate the compositional profile of our biomass. So we take one representative carbohydrate, we take a couple, for now it's one, representative of carbohydrates, of course nanocellulose. We're going to take one protein as representative of all proteins. We're going to take one lipid as representative of all lipids, and we're going to mix them in different concentrations and subject them to different processing conditions, so that we can basically have a clean way of trying and studying the reactions between those classes of biomacromolecules. So that inspired a collaborative project that we actually have. It's a very large NSF collaborative consortium that we got end of last year. So hopefully, and I'm going to show you the first results actually out of that. It's only 2 slides, but I'm very proud of them. So let's see. My student Ian is leading this effort from our side of lab, where he is creating different mixtures with different compositions, different relative concentrations of each of those 4 building blocks, trying to simulate the different types of biomasses that we actually have seen this plastic behavior occurring in. So you will see that we have single cellular, this is cyanobacterium, this is a green microalgae, this is a brown macroalgae, and this is a green macroalgae. So these are very, very different. It's not just the composition. Nevertheless, we have seen that you can actually process them with heat and pressure and get useful properties and thermoplastic behavior without extraction. So we know that the mechanism definitely happens in all 4 of those, regardless of their differences. So we are trying with this model system. It's the version 1, the results that I will show you today. But we now increase the complexity as we move forward. So we chose alpha cellulose as our high molecular weight carbohydrate, even though we only have it in 2 of these examples, or 3 of these examples, so we don't have it in spirulina. BSA is going to be the protein, the representative protein. It actually falls the molecular weight. It's about 56, 58 kilodalton. It does fall within the range that we have in our natural protein, but that's as much of the similarities as we can have. Sorbitol and stearic acid are going to be our small molecular weight carbohydrate and our lipid equivalent. So now we try and mix all of those in concentrations that are going to be similar to what we would find in biomass. And of course, here we're not talking about structure at all, just compositional similarities. So when we start mixing those materials together, you will see that cellulose, of course, if we don't do anything and it's dry mixing, I think it's speed mixing actually, before we put them in an extruder and then we put them in a hot press in the same, in a variety of conditions. We also do design of experiments there. But in some of the conditions, in all of the conditions, we see the same thing, where we have nothing is enough of these conditions to actually break up cellulose. It will not defibrillate. It will stay as agglomerated as it is, so it's a very, very large fiber. Protein is going to be forming a matrix around cellulose. So if you go with EDX, you will see your nitrogen all around this bulk matrix. So this is how we recognize that we have protein in a proteinaceous matrix. And then we will see also these characteristic crystals actually, not crystal-like, microcrystals. These are going to be the self-assembled crystals from our fatty acids, so stearic acid, which is what we would expect really, but they do migrate all the way up to the surface. Something that we haven't seen actually in real biomass, but we do see it in our simulation system. So our simplified system allows us to see that there is, that in this combination of materials, protein is playing the role of a matrix. Your cellulose fiber is going to be your structural fiber there, and we're going to see it in mechanical properties as well. They're going to be embedded, embedded in the matrix. These are going to be very, very hydrogen bonding prone interactions. They are designed actually for that, so of course they're going to interact very nicely. You won't see bad interfaces around these two. And then we did see the fatty acid assembling into microcrystals and migrating all the way to the surface. Now, in our mechanical properties, these are again flexural tests, and here we have the convention that you see here is the numbers correspond to weight fraction for or weight ratio for each of the components. C is going to be our cellulose, BSA our protein, sorbitol is going to be our low molecular weight sugar, a carbohydrate, and then stearic acid our fatty acid. So you see that we have a 3-to-5 ratio for between cellulose and BSA. That reflects to our spirulina concentration. And then we have a 10 and 10% of the small molecules. So we're trying to understand now the small sugars with the fatty acids, what is their role? In this case, the first data point that you see has both of the small molecules. The second bin has only sorbitol. The other has only stearic acid, so 20%, 20%, and here 10 and 10. And here you don't have small molecules. You only have cellulose and protein mixed together. So you see that our modulus and strength are highest when we don't have small molecules, when we have our strong fiber, cellulose, and some matrix, the protein working as a fiber composite, basically. And then when we start introducing the small molecules, in particular sorbitol, we see a classic plasticization behavior where we have increase in our strain at break. We have an increase in our toughness at the expense of our strength and modulus. That's how we use how you would introduce the effect that you would expect if you were to introduce an external plasticizer. We see the same behavior or similar behavior in our mixed composite, basically, but we don't see the same behavior with our stearic acid crystals, where you basically lose across the board. So you lose strength, you lose stiffness, and you lose extensibility. And that now starts making sense if we think about the picture that we saw before, because those crystals are strong, but they're not as strong as cellulose. And then the fact that they migrate all the way to the surface tells us that they would likely act as a defect, and that is, and that is what we actually see over here. That they function as a defect where they are reducing all the properties in our composite. So the first round of understanding, I guess, kind of makes sense. Then in this case, we are fixing the 10 and 10% small molecule concentration, and we are going to be changing the ratio between our protein and cellulose. So all the way to the left, we're going to have only protein, no cellulose, and your small molecules. All the way to the right, we're going to have just cellulose, no protein. Decreasing protein content. Yes, here you only have protein, here you have only cellulose. So maybe as you would expect, when you just have cellulose, your material, and the small molecules, your material is going to be very, very stiff and stronger than if you were to have just the protein. So the two binary, the two extreme concentrations are as we expect. Then we start decreasing protein content, and we start seeing that there are some compositions, in particular these two, where we have the smallest amount of protein mixed with cellulose, where we actually have some synergistic behavior. We are able to have higher strength than if we just had cellulose or protein. We are able to exceed that and be comparable in this case for the modulus. So primarily the strength is what is though influenced, and it does make sense because even in a fiber composite point of view, you do need a matrix to transmit the load between your structural fibers, and you should be expecting to see the difference in the strength rather than the modulus. So even if this was not a natural composite, it would somehow make sense. Here we're laying down the flexural properties for four different types of biomasses that we're trying to understand, to simulate. And you will see that we laid them down in order of decreasing protein content. So this is going to be the material that has the highest amount of protein, 65%, and then we're going to be lower and lower and lower amount of protein. So you will see that as our protein amount decreases, we do follow a similar pattern, at least in terms of our strength, where we would be expecting to see some similarities, even though that is very, very crude. And if you are critical, and you should be saying, okay, it's not just the protein concentration that you're varying here. Of course, here you also have carbohydrates and higher molecular weight and tissue connectivity. And if you were to think of that, I would tell you, yes, you're absolutely right. But now we are laying them down in a protein concentration, and that does make sense. So the reason why I wanted to remain quite vague or not read too much into the FTIR and XPS, especially from a real biomass, is because finally we do have a collaborator that is able to do molecular dynamic simulations for us. So then I don't have to pretend that I can measure a hydrogen bond amount from an FTIR, because they can actually simulate that. So our collaborator, Kayla Sprenger from University of Colorado Boulder, set up the system with her student Travis. They set up the system for our model, our synthetic analog, so that we can start understanding these before we actually go to the real biomass. So here they start from a dimer of our protein. This is BSA protein that they are modeling. And these are the FTIR results that we took, experimental results, powder, and then a pressed material. So this is a self-bonded material, and you can see that our hot pressing does change the alpha and beta conformations, as well as random coil, well, not really random coil, but the alpha and beta ratios. Even in pure protein, pure BSA, we do see a change. And then you see a bunch of different columns over here. Each of these columns represents a couple of months of work for poor Travis, because he had to go through all of these iterations to actually find a conformation that is, this is the last, I think, conformation, that is, or breakdown of alpha and beta sheets that is somehow close to what we actually experimentally measure. BSA is the most studied protein. You have models. If you want to do molecular dynamic simulations, it's the most well-studied protein you can actually find the crystal structure for. But this is the crystal structure that you will find in literature. And this is the native state. This is how it exists actually in whatever animals we extracted from. Bovine, I guess. It's in the name. But this is how it exists in nature. But this is what we buy from Fisher. So it's not the same thing. So actually, for a while, they were modeling this, and the results were not making any sense. They were not matching what we saw experimentally. And then we started seeing that this was the problem. So what we are starting from here is not perfect. It's not exactly the same as what we measure, but at least is close, and it does allow us to see similar trends. So now we know what the before and the after looks like from the experiments. We're trying to match that with the simulation. So then they're running a variety of different simulations of subjecting heat and pressure to this dimer. And then we're trying to see multiple things. First of all, what's happening to the conformation of our protein. So here you see a heat map. This is a residue map. So you see the first molecule and the second molecule, and you see which of the residues of your protein actually come together, closer together, are the ones that are going to be in the red color. And the ones that are in blue are the amino acid sequences that are pushed further apart as you hot press the material. So you do see systematic changes. There is this block of the residues, the 2 alpha residues that are between 250 and 350. They correspond to actually the hydrophobic domains, and they are the ones that are pushed further apart. Everything else comes closer together as you hot press the material. Then if we take the FTIR of this, and we have the powder over here and the pressed material over here, you see a very characteristic reduction, and this is normalized to one of the peaks. I should know which one the peak is normalized. Nevertheless, you do see a reduction in your amide 1 peak, and that in conjunction to the residue proximity maps, show us what people in biology have already reported as an aggregation mechanism that is dominating loss of function in a specific type of proteins, where they are relating that to some diseases. Loss of function in proteins, in living proteins, of course, leads to some diseases. So for them, it's a very bad thing. For us, we think that this is explaining, basically, this aggregation mechanism. We think that explains the matrix formation that we see in our plastics and our synthetic analog. So then if we start putting everything in the system, where with your purple, you see your protein, again, just a dimer, so only 2 molecules. You understand, these simulations have to be very tiny. The green is going to be our cellulose. The red is going to be our stearic acids, or fatty acid crystals that we will see their self-assembly. And the orange is going to be our sorbitol. So then we will start seeing, as we subject the system to heat and pressure, we will start seeing that indeed we should expect some crystals to form. This is actually confirming the SEM images that we had. So stearic acid will not interact with anything else but with itself. It will self-assemble. Then our sorbitol is going to go everywhere, plasticizing the matrix. Actually, not everywhere, primarily within the cellulose domain, even though there are some that interact with the protein. And then what we can extract from this simulation is the amount of hydrogen bonding between the different macromolecules. So we don't have to see all of that. We can just see this. This is the composite where you have both protein, cellulose, and the 2 small molecules. And if we just see only the 3 blocks that I have highlighted, if we start from the yellow is the amount of hydrogen bonds between our protein and our cellulose. So you see that before we hot press, just in the powder, when the the two substances are in proximity to each other, they will have a certain number of hydrogen bonds. When you do subject them to heat and pressure and to conditions similar to what we saw before for the real biomass, you increase the number of hydrogen bonds between these two. Then we also see another interesting fact. The sorbitol and the cellulose are going to be interacting with themselves, actually giving significant proportion of the hydrogen bonding amount, even before the hot press. So this, of course, if you know the structure of sorbitol, and you see all of the pending hydroxyl groups, and then you have cellulose, you would think, of course, they will click together, and and they will form bonds. Nevertheless, it's nice to actually see it, and to see that this is not disrupted, neither enhanced, actually, by the hot pressing function. And then the other thing that we see is that the BSA with the sorbitol also interacts. So sorbitol does not only interact with cellulose, though primarily it does interact with cellulose. So that's just the first glimpse. Hopefully, yes, all right. Hopefully we're going to have better results soon. This is the first, I guess, collaborative approach. So then if we go back to what we can do with this and why we cared in the first place, we cared because we are trying to see that if whole biomass can basically function as a new class of thermoplastics for us. What do you do if you want to change properties of thermoplastics? You introduce nanofillers to modulate the properties that you get, so that instead of one performance, you have multiple. So in this case, we chose nanoclay, which we know, this is montmorillonite. We know that it will interact favorably with the, of course, it will have hydrogen bonds with all the biomass. The abundant hydroxyl groups that you will find in spirulina. So this is just the spirulina biomass, and this is up to 5 or 8% nanoclay. So we can see that you have room to improve the properties further. I don't think that the properties are that impressive by themselves. The point that we're trying to make over here is that you can use it as a matrix as well. So you can use pure cells, whole cells, without any extraction or no other processing as a matrix. Then if you remember that we said that these are extremely brittle materials, if we wanted to extend a little bit more their flexibility, we would have to, and without changing biomass, because you could just choose a different biomass, but if we didn't want to do that, we can introduce an external intended plasticizer. In reality, it's actually functioning more as a binder rather than a plasticizer. Here we're introducing sorbitol back into spirulina as a filler material up to 38%. And you can see that at the highest concentration, we are able to, of course, at the expense of stiffness, not really strength, but the expense of stiffness, you can have some flexibility. And just for us to understand what 2.5% means, because I don't know if you can tell what 2.5% means. This is a filament that is extruded out of our extruder with a 30% spirulina, so that we can see that indeed this is no longer the brittle. If you were to see the one without any filler, you would not be able to do anything. In 0.5%, it's gone. So you can play with that. You can compression mold or transfer mold, or you can do machining, or you can do laser cutting with those. So we are demonstrating that you can actually use that as a regular plastic. One of the good things, of course, when you're using biomass and charring biomass in this particular case is that it will have self-extinguishing properties. So when you take it out of the horizontal flammability test, it will self-extinguish, it will stop the flames. Of course, this is a PLA filament, the same size, so everything is the same dimension, so that we can do a one-on-one test. And then why we call this a thermoplastic is because you literally can mechanically recycle it, and these results are coming from beam, coffee grinder, powder, and then hot pressing multiple times. And we can see the mechanical properties holding up. You do have some losses, and we're actually going and characterizing each iteration so that we can see how much of the covalent bonds are we losing with every processing step, and how much are actually recoverable and dynamic. So hopefully next time we'll have some answer here. Of course, if you have these types of materials, you can expect, this is just protein, carbohydrates, and lipids. Of course, it will decompose like food waste. This is going to be the positive control. The yellow is a banana peel. So you can see that we have the same kinetics in the pure spirulina beam as a banana peel. This is just in a burial test, and this is in an ASTM equivalent where we have the evolved CO2 to please the reviewers. But I think this should be strong enough. Nevertheless, regarding the strength of the materials, this is properties in compression are way better than in bending, so that you can see this is a 1 by 1 inch by 1 inch beam cube of pure spirulina. There's nothing else in there. It's about 70 MPa in strength. I don't know what that means, so I asked my students to actually do a demonstration. I asked for a human pyramid. They gave me just one person on the beam, and then they drove a car over it. So at least then we know that it is strong enough to hold the car. So that is that. And then we have demonstrated the same thing with invasive species as well. This is actually overblooming brown seaweed that is very problematic in the Gulf of Mexico, maybe far away from here, still very big environmental concern. We have shown that you can do the same. You can 3D print. I think I have to, and yeah, and all of the different colorful bubbles that you see over here, different compositions that we were able to do with our collaborators. So we have some indigenous species from Hawaii. We have some that they are grown, some that they are harvested by non-profit organizations because they are problematic. So we can see that you can use the same methodology, more or less, to populate more areas in that. And maybe to close up, this is just maybe half or 60% of what we do in the lab. We actually do other things as well. We do work with adhesives that are coming from biomass. We are studying nanoparticle formulations, of course, bacterial cellulose films, colloids, hydrogels, foams, and biochars. These are going to be pure cellulose. The rest are going to be mixed biomass. And then I think I don't have time to talk about cement at all, but if you are interested, I can also talk to you about our structural materials later. So that would be all. Thank you. Just slow.
Thank you so much for that exciting and enthusiastic presentation. And I didn't mean to rush you. Maybe people have questions about the cement as well. So we have 10 minutes for questions now. Thank you so much, Eleftheria. Do we have questions from the audience? I'll bring the microphone around so that it is captured in the recording. But yeah, Jaya?
Thank you so much. I have heard a lot about you over the news, but now I see what you did. So I have one basic question, one little bit more technical. I'm a biologist, so I really want to know what is the PHB content here as a biopolymer? PHB content here, if I use
PHB, less than 10%. I want to say 5%, but it's less than 10. I don't remember exactly how much.
Okay. My second question is, you did use the heat and whatnot, but what will happen if you add the inorganic salt to it because of the protein and you are talking about the carbohydrate?
Now, that is a super exciting question. Both of them were very interesting because you do have PHAs in spirulina, but and we were thinking how to what extent this would help, because it's a polyester, it would help the flow, but it is very, very low. So I really don't think that this is a decisive factor here. And we don't have it in the other biomasses. So that's why we kind of don't think that this is a very big part of the answer. pH, from the protein content alone, you can do beautiful things with different salts. You can induce cross-linking, of course, with your chargeable polymers, not your cellulose, but definitely from the proteins. The proteins that we have are in a denatured state by themselves as we get them. So I don't know to what extent you can change conformations by enabling complexations. But if we were to look, when we haven't unfortunately dove into that, but I really think that if you have the mean solution and then you can play with your pH and salt concentration and all of that, it should be that you can induce important changes. We have not looked at that. But great question.
Great presentation. Thank you for the talk. In one of your slides, you showed the mixtures between protein and cellulose, and you showed that there was a synergy in the strength. Can you elaborate on what kind of interactions do you think are forming that? What's the source of the synergy?
Yes. Oops, the next one. Here.
Right.
Yes. So even from our simulations, we know that we do need a certain amount of matrix to do the load transfer between the fibers themselves. If you have just the fibers by themselves and the small molecules, it's not enough. So your matrix, there is no matrix. Therefore, you do need some protein. But we haven't actually identified what is the sweet spot or a maximum, because you see that the last point is a 20%, 2 to 8 by weight protein to cellulose. I actually think you don't need that much. I think you would go higher if you have less amount of protein in that particular composition. But we wanted just, this is just to scan effectively the space. It's not to identify the maximum. I think the synergism comes from the fact that you form a matrix that is cohesive and you don't have poor interfaces. So the fracture always happens in the matrix, actually. It doesn't happen between the fiber and the matrix. If you had the carbon fiber composite, for example, your weakness is always the adhesion between the fiber and the matrix. In this case, you don't have that problem, and your fracture happens through the matrix in itself. So the protein is the weak link, yet you do need some of it.
But are you creating covalent bonds? Is there?
No, but I shouldn't say no. Not that we know of. So first round of simulations is only for secondary interactions. So we have measured the amount of hydrogen bonds only. Now we're running the REACT-FF, where it's a different framework, so that you can change the covalent bond changes. You can measure the covalent bond changes as well. We have not measured that. And if the people are doing molecular dynamics here, we can discuss the pros and cons from each one, because I'm not an MED person, and I really wanted to push our collaborator to do both. But people have very, very strong feelings as to how to do the force fields in the one case and in the other. Nevertheless, I do have the same question as you, so we do need an answer. And so far, we don't think that there's going to be covalent.
Because you may have Maillard reaction. If it's done under heat, you might get Maillard reaction.
But these are quite mild conditions. Like in this particular case, we're pressing at 80 degrees C, and I want to say 1 MPa. So that is a good suggestion, absolutely. And I do think that there's also fiber level interactions, not just the hydrogen bonding. But let's see if we have Maillard reactions, maybe.
Thank you.
Thank you.
Do we have other questions for Eleftheria? Yeah, Laurel.
Thanks very much. So seeing as you're relying on extensive hydrogen bond networks, where hydrogen bonds are very dynamic, in particular in these systems, I'm interested in aging effects.
Yes. These are very, very good. So in that program that I presented to you, the collaborative, we do have aging in terms of cycling, heat pressure cycles, humidity cycles, UV also, because we may have also some degradation of these materials with light as well. So it is part of the 4-year project to also assess the cyclic behavior or stability, I guess, in different conditions. Definitely the losses and form and reform events of hydrogen bonds, I think, are very delicate to attempt to quantify. But we're going to give it a try and see.
And then kind of related to that would be shear hardening effects?
Let's see. I'm trying to remember, because we did have in our last meeting, somebody already presented something, but I am very hesitant to tell you any results because I really don't remember them. But we are looking into it as well. I should have studied better before coming. But it was last week.
I had a quick question. The spirulina powders you showed looked like they were spray dried. Do you know how those powders are made?
I think these are either freeze dried or air dried, actually.
Okay. And they form those spheres, because I was just wondering if drying conditions or processing, even before you get to that, could have a additional beneficial effect on the composites.
This is a good comment. Some processing conditions do matter a lot. So for the seaweed that we have, that actually we control all the, because we get it green.
Yeah.
So that one we have done freeze drying, We have done cryo milling and then sun drying, I believe, and there are differences that you will get, absolutely. Luckily, we got like I think a 10-kilo batch, so we wanted to fix, you understand very well how you also have seasonality in your feedstock, so you know.
Yeah. Great. Okay, well, I'd really like to thank the audience for being here today with us. A special thanks to Kelly and Cindy, who helped with organizing the event, and the BioProducts Institute in Orlando for sponsoring us. And of course, please join me in thanking our speaker for a wonderful presentation. Thanks.
Thank you. Thank you.